Firebird Biomolecular Sciences solves problems in nucleic acid diagnostics, not by combining off-the-shelf natural DNA, but by reinventing DNA itself to improve multiplexed and even highly multiplexed PCR assays. Ideally, during PCR, DNA primers should solely bind to the target sequence, initiating the primer extension that duplicates that sequence. DNA replication follows, yielding billions of copies of the target. However, PCR is rarely ideal. Primer dimers arise when primers bind to each other with sufficient affinity to initiate unwanted polymerase extensions, forming the wrong or off-target amplicon. These undesired amplifications waste reagents, lower assay sensitivity, and may lead to false positive or false negative results. Firebird has solved all primer-dimer-related problems with its self-avoiding molecular recognition system, or SAMRS. SAMRS inserts reinvented nucleotides into the primers, which bind only to the desired DNA or RNA target and not to other SAMRS primers. These four reinvented SAMRS nucleotides, A star, T star, C star, and G star, bind to their natural DNA complements with two hydrogen bonds. However, SAMRS to SAMRS pairs have only one hydrogen bond, which is insufficient to stabilize the double helix. Therefore, SAMRS primers ensure that PCR reagents produce the desired target amplicons and not side products, rendering primer dimers a problem of the past. Today, even for a low multiplex PCR assay that seeks to detect only a few targets, it can take months of painstaking development to obtain acceptable performance. Unpredictable and unintended side products, such as primer dimers, can also leave those molecular assays less robust, with slight variations in lot-to-lot manufacturing, resulting in significant quality control issues. But with SAMRS primers from Firebird, new PCR assay development becomes much more predictable, since optimized single-plexed assays can be combined into one multiplexed assay with comparable individual assay performance without significant optimization. Most PCR applications intended to detect 10 to 100 or more targets are split into multiple reaction vessels. Each vessel has one set of primers and one probe for each target, and a pre-amplification step is often required to provide enough sample for every amplification. The reduced primer-dimer formation with SAMRS can substantially improve the pre-amplification step. Currently, most multi-parallel detection tests are limited to less than 25 targets, but based on the large multiplex PCR amplifications enabled by SAMRS technology, it will be possible to conduct assays with at least 100 targets, which is impossible with standard reagents. Many major diagnostic applications, such as antibiotic resistance and cancer therapy selection markers, require a large number of targets. In some cases, the specific target need not be identified. For example, antibiotic resistance testing must seek 50 or more genes that confirm resistance to each antibiotic. If any of these markers is present, physicians must change the patient's prescription, even if they do not know which particular genetic alteration has been detected. Only SAMRS primers can provide reproducible results for assays at this high level of multiplicity in one reaction vessel. The only other alternative assay methodology at this level of multiplexing requires next-generation sequencing, which is suitable only for centralized laboratory settings. SAMRS enhanced assays, however, can be performed on any existing PCR system, installed in both centralized and decentralized testing sites, even on those systems which are currently limited to just a few single or low-plex assays, opening the door to previously unimagined opportunities. Firebird Biomolecular Sciences has improved upon nature by reinventing the building blocks of life to deliver multiplexed PCR diagnostics without limitations. Firebird Biomolecular Sciences, igniting innovation. Learn more at firebirdbio.com.